let others also join. Okay, 39. I think today's attendance is less, so let's start, okay? Or shall we wait for some more time? Hello? Hello?
Yes, ma'am.
Shall we start or shall we wait for some more time? It's less, 41. Only 41 have joined. Okay, so let's start. Okay, let them join in between. So today we will discuss about synapses. Hello, is my slide visible?
Yes, ma'am.
Hello.
Yes, ma'am.
Yes, ma'am.
Okay. So in the previous class we have discussed about different types of cells in the nervous system. What are the cells present in the central nervous system, and what is the difference in the characteristic of the cells present in the peripheral nervous system. And today we will discuss about synapse. So in the nervous system, what is the meaning of synapse? So in the nervous system, synapse is a structure that permits a neuron to pass an electrical or chemical signal to another cell. Means it is a means. It is a means. It is a structure. It is a means which is present as a structure and which permits the transmission, okay, which permits the transmission or which permits the passage of an electrical or a chemical signal to another cell. So based on the parts involved at the spot, based on the parts involved at the synapses or based on the parts of the neurons involved at the synapses. The synapses can be named different ways. Say exosomatic means here the body of a neuron synapses with the axon of another neuron, means an axon of a presynaptic neuron synapse with the body, that means the soma. Okay, that means here this is the body of a neuron and this is the axon terminal of another neuron. The axon terminal of one neuron is synapsing with the body, that is the soma of this postsynaptic neuron. Or it may be exodendritic. That means here the axon terminal synapsing with the dendrite. Exonic means this axon synapsing with the another axon terminal with the another axon terminal. Or exospinous means the spines means what are those? These are the spines means these are the fine projections present at the dendrites. So there synapses with the spines, then that kind of the synapse is exospinous. And it can also be a neuromuscular junction. So at this time, the junction is not between nerve and nerve, but it is between a nerve and a muscle. Nerve and a muscle. Okay, so that is neuromuscular junction. The synapse, if it is nerve to nerve, it can be the different portions of the of a neuron, and the neuromuscular here, one presynaptic neuron, presynaptic structure is a neuron, and the postsynaptic structure is a muscle. Okay. So it can be in different ways. So when we discuss about the neuromuscular junction, the neuromuscular junction is also, hello, yes? Wait, okay. Let them try again. I'm on that page. Let them try again to join. Is it done?
Yes, ma'am.
Somewhere. Okay. Yeah. So what I was telling, the synapse means it's a structure that permits the passage of an electrical or chemical signal from one neuron to another cell. So the synapse may be between two neurons or it may be between a neuron and a muscle. So that kind of the synapse involving a neuron and a muscle is called a neuromuscular junction. And also depending on the site of the or depending on the sites of the neuron involved, a synapse can be exosomatic, it may be exodendritic, it may be exonic, or it may be exospinous. Okay, so the meaning is synapse means it's a structure which permits a neuron to pass an electrical or chemical signal to another cell. That another cell can be a neuron or it may be a muscle. Okay, so these different types of the synapse based on the part of the neuron involved in the synapse. So here, this is the same figure which I have shown in the previous slide. Here, the synapse is exonic, exodendritic, or exosomatic. Okay, or it can also be spinous. Means if the axon terminal is synapsing with the spine. Then it can also be a neuromuscular junction. In case of the neuromuscular junction, the presynaptic structure is a neuron and the postsynaptic structure is a muscle. Okay, so here in this slide, in the previous slide, I said that a neuron passes a electrical or chemical signal. So this electrical signal from the presynaptic neuron will be passed on to the postsynaptic neuron or the postsynaptic muscle structure. Okay, so in that, just 1 minute. But just 1 minute. Okay? Just 1 minute. So what I was telling, that message or that signal can be in the form of the electrical or chemical signal. So what is that? Electrical means the message which is brought by the presynaptic neuron will be in the form of the electrical. Means there will be no involvement of any chemical substance at the synapse. Okay, but in some cases the transmission or the passage of this message is going to be in the form of a chemical. Okay, so that chemical, in case of the chemical synapse, say I come to the term chemical synapse. So this chemical is going to find its receptors on the postsynaptic structure. It can be a neuron or it can be a muscle. So in case of the neuromuscular junctions, the chemical substance will be released from the presynaptic neuron. That chemical substance will come out. That chemical substance will be called a neurotransmitter. That neurotransmitter will come out and they will find their binding sites. They are called the receptors. Those receptors are present on the postsynaptic structure. So in case of the neuromuscular junction or in case of a chemical synapse with a neuron, that neurotransmitter will find the receptor present on the postsynaptic structure. They will bind. Once they bind, this postsynaptic structure is going to respond. Okay, depending on the type of the neurotransmitter. So earlier, the synapse before the binding of the neurotransmitter to the postsynaptic neuron, we term it as a resting synapse. And once the chemical substance has bound to the postsynaptic neuron, that is now called an active synapse. Okay, so at this point, what you have to understand is that synapse can be electrical or it can be an chemical synapse. But in case of the electrical, there is electrical. Hello?
Ma'am, some students are still trying.
Yes?
Some students are not able to join the class, ma'am.
Let them try again once. I'm on that page. I don't know why. Maybe due to internet. Let them try. I think one has joined just now. Is it done? Oh, now 49.
Thank you, ma'am.
Okay. Okay, let's continue. So, at the synapse, different locations, different portions of a neuron can involve, okay? And before the and the synapse can be electrical or it can be chemical. So in case of the electrical, what happens that we will discuss again. But in case of the chemical, the neurotransmitter will be released from the presynaptic neuron and bind to the receptors in the postsynaptic neuron or postsynaptic structure, maybe a muscle. Then that synapse becomes active, okay, active synapse. Earlier, before binding, before release of the neurotransmitter, that synapse is a resting synapse. Okay, so this figure is showing an axon terminal. Axon terminal having vesicles containing neurotransmitters. The neurotransmitters are present over here. These neurotransmitters, you know, where they are synthesized, they will be synthesized in the cell body, in the body of the neuron. And once they are synthesized, they will be brought down the axon, okay, and down the axon, and during that passage, they will get matured, okay, that process will complete. And by the time they are mature enough, they will be present in the form of vesicles at the axon terminus. Those axon terminus are called the N buttons. Okay, so those N buttons will be having the neurotransmitters in the form of vesicles. Okay, so there is a gap between the presynaptic neuron and the postsynaptic neuron, and that gap is called the synaptic cleft. Okay, or and by the time these neurotransmitters are released and bound to their receptors, that means they are going to open different other channels in the postsynaptic structure that we will discuss later on. Okay, so once those neurotransmitters release onto the target cells, they are going to open different other channels, and that target cell will be having its receptors at the plasma membrane. Okay, target cell plasma membrane having the receptors. So neurotransmitters, when they bind to their receptors, not all these neurotransmitters have the ability to bind. Some get accumulated here without binding, and those who are left without binding will be deactivated, or they will be destroyed. Okay, so different other chemicals or different other enzymes will be involved to destroy these leftover neurotransmitters. Okay, so that part we will come again later on. So this figure is just showing the synapse, say here exodendritic, okay, and this structure, yeah, another neuron synapsing at exosomatic. So this neuron is a presynaptic neuron. This neuron is a presynaptic neuron, and this neuron is a postsynaptic neuron to this neuron or to this synapse. Okay, so this is the postsynaptic neuron for this synapse. Okay, this neuron is a presynaptic neuron for this synapse, exoaxonic synapse. So this is also a presynaptic neuron. Okay, if this neuron synapses with another neuron, then it will be this will be a presynaptic neuron, and the another one will be the postsynaptic neuron. Okay, so the central nervous system synapses, what are that? So these synapses, the information is transmitted in the central nervous system, mainly in the form of nerve action potentials, called simply nerve impulses, through a succession of neurons, one after another. So how these message, how these signals, okay, are transmitted? They are transmitted in the form of nerve impulses, and these are nothing but the nerve action potentials. So this neuron will be transmitting the impulses in the form of action potential to this neuron. This neuron will be transmitting its impulses in the form of action potentials to this neuron. Okay, so each impulse may be blocked in its transmission from one neuron to the next. That means the message, the impulses with this presynaptic neuron is brought to here, it may be blocked at any instant. It may be blocked at any instant. How it is possible? Suppose if we do not allow these neurotransmitters to combine or to bind with these receptors, then also we can block. Is it possible or not? If we remove these substances, if we put certain chemicals near around this synapse, which do not allow the binding of these neurotransmitters, either by destroying these neurotransmitters, or the substance which are present over here bind to these receptors, but stopping the binding of these neurotransmitters. That way also it can block. Okay, so the impulses may be blocked in its transmission from one neuron to the next, or it may be changed from a single impulse to repetitive impulses. So these you may not understand now, okay? So later on you will come to know that the impulses which is coming in single form, it may also be changed into repetitive impulses, or it may also be integrated with impulses from other neurons to cause highly intricate patterns of impulses in successive neurons. So in this neuron is receiving a signal from here, a signal from here. If this signal and this signal is of similar type, this neuron is going to get more of the effect. If the signal brought here is empty, or it is blocking the signal brought by this neuron, then this neuron may not get any effect. Is it possible or not? So this neuron may receive many signals, and those signals may influence the activity of this neuron in different intricate pattern. The meaning is that, okay? So if it is of stimulatory type, and this is also a stimulatory type, then this neuron, postsynaptic neuron, will be stimulated. If this is stimulatory and this is inhibitory, these two may cancel. Then this neuron may not respond at all. Okay, so that is the meaning of this. So it may be integrated with impulses from other neurons to cause highly intricate patterns of impulses in successive neurons. Okay, so based on this mechanism involved, the synapses can be a chemical synapse, or it can be a electrical synapse. So almost all the synapses used for signal transmission in the central nervous system in the higher animals are of chemical synapse. So there is more, in most of the situations, there is involvement of the chemical substance. So those chemical substances are nothing but the neurotransmitters. Okay, so this classification is based on the mechanism. The earlier classification say exonic, exodendritic, exosomatic. That is based on the site, based on the sites of the neuron involved at the synapse. Okay. Okay, so in the higher forms of the animals, almost all of the synapses present in the central nervous system, they involve chemical synapse. Okay, so what are the characteristics of a chemical synapse? So a chemical synapse is characterized by, first point, there has to be release of a neurotransmitter. Okay, who will release this neurotransmitter? The neurotransmitter will be released by the presynaptic neuron. Okay, then what this neurotransmitter is going to do? This will bind to the receptor located in the postsynaptic cell. Means, then you see in this figure is showing a portion of a chemical synapse involving one portion. This is the presynaptic neuron and this is the postsynaptic neuron. This is the axon terminal and this is supposed a soma of a neuron. That means the body of a postsynaptic neuron. The neurotransmitters will be present in the form of vesicles at the axon terminal. Okay. Once those neurotransmitters are released from the presynaptic neuron, the receptors present on the postsynaptic neuron will be ready to bind with those neurotransmitters. Okay, then those neurotransmitters will bind to that receptors on the postsynaptic neuron, and now there will be change in the permeability of the membrane of the postsynaptic neuron. Have you discussed about this action potential with Sir MC? Hello? Have you discussed about the action potential? So, how does the action potential develop? You just tell me in brief. How the action potential develops?
Depolarization of membrane.
Yes, depolarization of membrane. Earlier it was polarized. That means the membrane was having more of the negativity inside, isn't it? Yes. So earlier, before this synapse, okay, before this synapse happened, this membrane, the postsynaptic structure, okay, so at this time it is a soma of a neuron. The membrane, okay, will be maintaining the potential difference of negativity inside. Now this neuron is polarized. That means polarized inside, that means there are more negativity inside the membrane of this postsynaptic neuron. But by the time these neurotransmitters go and bind to their receptors, now this postsynaptic structure, the postsynaptic neurons, permeability to different other ions, to the positive ions or the negative ions, will change. So, who brings this depolarization? You said that depolarization, action potential develops due to, yes, due to the sodium ion. So now this membrane will be more permeable to sodium. So the sodium will enter more to that postsynaptic, to the across the membrane. So the membrane will get depolarized. Earlier it was polarized, and now due to the influx of more and more sodium ions inside, because outside the cell, outside this neuron, more of the sodium ions are present in this in the extracellular fluid. But inside the cell, the neuron is having less of the sodium ions. So the sodium ions will influx. They will come inside more. Now this neuron will be depolarized. From the negativity, it will change to positivity. So as the membrane potential rises from polarized state to say minus 90, minus 70 or to minus 90, then it will be depolarized. Okay, so when the depolarizing potential, okay, when the depolarization occurs, if the potential, the membrane potential reaches a threshold value, there will be an action potential. Okay, so this action potential is now going to pass on throughout the length of the postsynaptic neuron. Okay, and that change is brought by the binding of the neurotransmitters at the receptors in the postsynaptic neuron. And that kind of the synapse is called a chemical synapse. Okay, so what are the three main characteristics? One is the release of a neurotransmitter, next is the binding of the neurotransmitter, and depending on the nature of the neurotransmitter binding to the receptors, these postsynaptic neuron may depolarize or it may further repolarize. Okay, if it depolarizes and if the membrane potential reaches a threshold value, the postsynaptic neuron will be having an action potential. So the three steps are there: release of neurotransmitter, binding of neurotransmitter to a receptor located on the postsynaptic cell, and these will either bring out the excitation of the neuron or inhibition of the neuron, or modify its sensitivity in some other way that it may affect the postsynaptic cell in a wide variety of ways. That means how this postsynaptic neuron is going to respond. So that kind of the synapse is a chemical synapse. Is it okay? Will you be able to write the characteristics if someone asks what are the characteristics of a chemical synapse? I hope so. Okay, so that is a electrical synapse. So this is very less in the central nervous system. They are more important in generation of action potential, and they are characterized by connection of presynaptic and postsynaptic cell membranes by channels. Channels means these are some structures present across the membranes which allows the passage of the ions. So the channels are capable of passing electrical current, causing voltage changes in the presynaptic cell to induce voltage changes in the postsynaptic cell. Okay, so across the membrane of a neuron, in the presynaptic neuron, there will be certain structures which allows the passage of the different other ions from inside to outside or from outside to inside. And due to the passage of the ions, the positive ions, if it moves inside more, then there will be creation of positivity inside the cell. Then that is going to lead the cell to a depolarization state. Okay, and the same way may happen in the postsynaptic cell also. So depending on this type, okay, depending on the type of the neurotransmitter involved here, this postsynaptic neuron may either be excited or that may be inhibited. Okay, the potential difference which develops on this postsynaptic neuron may be either excitatory or it may be inhibitory. So depending on that, the postsynaptic potential may be called as excitatory postsynaptic potential, or that synapse which creates the excitatory postsynaptic potential on the postsynaptic neuron, that is excitatory chemical synapse, or it may be inhibitory postsynaptic inhibitory chemical synapse or inhibitory postsynaptic potential. Do you get my point? This potential which develops at the postsynaptic neuron may be excitatory or it may be inhibitory. Do you have any confusion here?
No, ma'am.
Okay, I will proceed. Okay, so the synapse, the chemical synapses may be excitatory chemical synapse. That excitatory chemical synapse is also called excitatory postsynaptic potential, or it may be inhibitory chemical synapse, that is also called inhibitory postsynaptic potential. Okay, so in the excitatory chemical synapse, the postsynaptic response to the neurotransmitter is a depolarization. You understand? That means the membrane earlier was polarized, means it was negatively charged, but after the synapse, after the neurotransmitter has bound to the postsynaptic neuron, okay, at their receptors, then the membrane potential changes from polarized state to the depolarization state. Okay, that may happen only if the more and more of the positive ions move inside the postsynaptic neuron. Okay, the postsynaptic structure. Okay, postsynaptic cell, bringing the membrane potential closer to a threshold, and the usual effect is to open postsynaptic membrane ion channels that are permeable to sodium, potassium, and other small positively charged ions. So in case of the excitatory chemical synapses, what happens? This allows the more permeability to sodium, potassium, and other small positively charged ions. But at this time, though the membrane is more permeable to sodium and potassium and other small positively charged ions, sodium concentration is higher outside the cell, while the potassium concentration is higher inside the cell. Do you know the concentration difference of sodium and potassium inside and outside of the cell? Do you know? how much, huh?
Ma'am, sodium
Yes,
concentration
Yes
ma'am.
or
10 to
no?
the power 7 times higher
Yes, ma'am.
than potassium concentration.
Sodium goes
Where?
out from the membrane during depolarization.
My question is, whose concentration is higher inside or outside? Say, talk about sodium. Sodium's concentration is
Inside,
higher inside or higher outside? Who?
Outside.
inside,
Where?
ma'am.
Normally. Sodium is higher
Inside, ma'am.
outside,
Outside.
while potassium is higher inside, right? Okay, so it is for sodium, it is more, it's 10 times higher, okay? And potassium is around 11 to 14 times higher inside, okay, if we compare with the outside. So we say sodium is an extracellular ion and potassium is an intracellular ion. Since the sodium concentration is higher outside, though the postsynaptic membrane is opening for sodium and potassium, sodium can come in, but potassium cannot come in because the potassium is already higher inside. Okay, so at this time, more sodium moves in while the potassium exit is not possible. So due to that, the cell gains more and more of the positive ions. Okay, so due to that, there is more influx of cations into the cell, and in most cases, it is due to sodium ions because the cell is having higher of the potassium inside and the potassium is lower outside, so it cannot send, it cannot get potassium from outside, but it receives more of the sodium. So due to that, the membrane changes from polarized state to depolarized state. Okay, so those kind of the chemical synapses are excitatory chemical synapses. Okay, so these ions then are now free to move according to the electrical and chemical gradients across the membrane. So there is both an electrical and concentration gradient driving the sodium into the cell. So what do you understand by this electrical and the chemical concentration gradient? What do you understand here? Do you understand electrical gradient? Yes or no? I assume no. What? Okay, I assume no. Okay, you don't know. That means electrical gradient means inside the cell before the synapse it was negatively charged. So this negatively charged, okay, this inside attracts more of the positive ion, and also sodium concentration is higher in outside and the lower inside. So there is electrical gradient from outside of positive ions and also concentration gradient for sodium from outside to inside. While for potassium, the electrical gradient is opposed by the concentration gradient. That means concentration is higher inside, though the negativity attracts the potassium, this concentration gradient opposes. Okay, and then it results to simultaneous movement of relatively small amount of potassium ions out of the cell and larger number of sodium ions into the cell. Okay, so thus the net movement of positive ions is into the postsynaptic cell, causing a slight depolarization. Okay. Then in case of the inhibitory chemical synapses, what happens? The potential change in the postsynaptic neuron is hyperpolarizing. Hyperpolarizing means from the negativity, it goes more to the negativity. Say minus 90 millivolts, so is the resting membrane potential, it will go further to minus 95, minus 100, minus 110, like that. Okay. So in this, what happens? At the inhibitory synapses, the activated receptors on the postsynaptic membrane open the chloride or potassium channels. And at this time, sodium channels are not affected. So now what will happen as the more and the more chloride channels are open, the chloride is also one extracellular ion. It will move more inside, and potassium channels are also open. So now potassium will move outside. So the cell will be losing the positive ions and it will be gaining the negative ions. So now at this time the cell will be more polarized. Alternatively, there may be no IPSP but rather stabilization of the membrane potential at its existing value. So this is another situation, okay? It may also happen. In either case, activation of the inhibitory synapse lessens the likelihood that the postsynaptic cell will depolarize to the threshold and generate an action potential. The meaning is that since the membrane is getting more towards the negativity, there is less chance to depolarize because the threshold potential is much higher. Okay, so the inhibitory chemical synapses will lessen the chance of creating an action potential, while the excitatory postsynaptic potential will increases the creation of an action potential. Okay, so when we plot the action potential curve, you know how the action potential curve is drawn. The resting membrane potential and then action potential. There is increase in the potential, then decrease in the potential. So this is one EPSP without reaching the threshold potential. Okay, so one EPSP is there. This is the change in the membrane potential in the postsynaptic neuron. Then potential is rising. Again, it is coming down. Okay, and it is another. This is the resting membrane potential in the postsynaptic neuron. Okay, going down, and there is one impulse. Due to that, there is one IPSP. It is inhibitory postsynaptic potential. So the membrane potential, instead of remaining at the resting state, it is more polarized. Okay, so in case if more EPSPs are given, okay, together one after another, then the potential may sum up and it may reach to the threshold value, and there is a creation of an action potential. But if there is one EPSP and another IPSP, then it may cancel each other. Okay, then many EPSPs, then they may add up and they may create an action potential. Okay, so in that way, this is one postsynaptic cell receiving many impulses from many presynaptic neurons. So this one presynaptic neuron, presynaptic neuron, presynaptic neuron, presynaptic neuron. Just imagine plus is indicating an excitatory and negative is minus is indicating an inhibitory input. So these plus ones, they will add up and they will minus this negative one. Okay, so these, depending on this synapse, this postsynaptic neuron will be having an EPSP. Depending on this synapse, this postsynaptic neuron will be having an EPSP. Here also EPSP, but depending on this synapse, this will be having what? An inhibitory postsynaptic neuron. Okay, so thus that whether this postsynaptic neuron will be either having an action potential or not will depend on the sums of all these excitatory postsynaptic potential and inhibitory postsynaptic potential. If these potentials are very, very less and this potential is very, very, if this potential is much bigger than the sum of all these, then this postsynaptic cell is not going to have any action potential. But if this is less and these are all big, then this postsynaptic cell is going to have an action potential. Okay. Do you get what we have discussed today? One is what is synapse, Another is depending on the site of the structures involved. Synapse can be named different ways, and depending on the mechanism involved, it may be a chemical or it may be an electrical synapse. Okay, and in most of the instances, it is a chemical synapse. The chemical synapse involves the release of neurotransmitter. After the chemical synapse, the postsynaptic cell is going to respond in the form of an excitatory postsynaptic potential or in the form of an inhibitory postsynaptic potential. Okay, and if the excitatory postsynaptic potential is big and if it reaches to a threshold value, then there will be creation of an action potential. So the synaptic cell may develop action potential or not, depending on the type of the chemical synapse happen. Okay, depending on the chemical synapse at different locations of that neuron. Okay, this we have discussed. 3 points we have discussed. Okay, you read. Okay, simple way I have discussed. So you read it. Can we end here? Do you want any further explanation? Shall we end here? Okay, let's end here. You read once, okay? Just for
Okay, ma'am. Thank you, ma'am.
you can leave. Uh,
Thank you, ma'am.